Good day, everyone, and welcome to Clinical Bacteriology Laboratory Session. Today, we will discuss principles of staining and the different stains used for identification and differentiation of bacterial cells. Now, in definition, stains po are chromogenic solutions that are essential in the microscopic identification and characterization of organisms. Now, the differences in the staining properties of the bacterial cell wall is the first step in microbial identification. When we say staining, now it is a process of coloring of colorless bacterial structures using stains or dyes. Now, this is used to identify microorganisms selectively by using dyes, fluorescence, and radioisotope emission. Now, the staining reactions are made possible because of the physical phenomena of capillary osmosis, solubility, and absorption of stains or dyes by cells of microorganisms. Now, the individual variation in the cell wall constituents among different groups of bacteria produce variations in colors during microscopic examination. And this is the most important part of bacteria that usually interacts with the stain or dyes employed in the procedure. Now, the objectives of the staining are the following. Firstly, is to determine the morphology of bacteria. Second, is to differentiate groups of bacteria. And the third po is to identify organisms with special structures, such as capsules, flagella, metachromatic granules, and others. Okay, let's now go deeper in the staining of bacteria and how staining of bacterial cells structure is utilized or done. So there are different types of affinity explaining this attraction forces. So these are your hydrophobic bonding, reagent cell interaction, reagent interaction, and other bonding such as your ionic, hydrogen, and covalent bonding. So these bondings happens within the biomolecules between the bacterial structure and the dye or stain employed. So why are stains po not taken up by every microorganism? So there are four factors for controlling the selectivity of microbial cells. So these are the following. First is the number and affinity of binding sites. Now the binding sites is very important po in the attachment of the different dyes or stains po used in the procedure. And alteration or the number of binding sites po and the affinity with the dye affects po the selectivity of microbial cells. Secondly, is the rate of reagent uptake. So there are different properties or chemical properties of stains or dyes. So some dyes are water-based, others are alcohol-based, and the like. Now, these properties or chemical properties of dyes must be in sync or in that it can or it must be soluble in what the structure po of the structure in the bacteria that you are staining with. So the rate of reagent uptake is very crucial in the selectivity of the microbial cells to stain. Now, thirdly is the rate of reaction. So this allows naman the interaction of the different reagents you employ in the procedure and um, the interactions of these chemicals may vary and may affect the selectivity of the microbial cells. And then the last one would be the rate of reagent loss. So this applies naman in staining procedures that needs differentiation or what we know as regressive staining. So the higher the reagent loss, no, the more chances or the more lower po, the more lower is the staining reaction. So as the lower naman the reagent loss, you allow the image no to have a better contrast and colorization po of the structure you are staining with. Okay. Now in this slide po shows you the different kinds of ionizable dyes used in staining bacteria. And these are the following. These are your basic dyes, acidic dyes, and neutral dyes. Now, your basic dyes are cationic dyes po with positively charged groups, which are pentavalent nitrogen, which adhere po to negatively charged molecules such as nucleic acids and proteins present in the bacteria. Now, examples of basic dyes are your methylene blue, your basic carbofuchsine, your crystal violet, your safranine, and your malachite green. Next po would be your acidic dyes. Now, your acidic dyes naman po are anionic dyes with negatively charged groups of carboxyl and phenolic functional groups that bind to positively charged cell structure. Now, examples of acidic dyes are your eosine, your rose bengal, and your acid fuchsine. Now, lastly, no, there exists a kind of ionizable dye which is called a neutral dyes. Now, when we say neutral dyes, the acidic and basic components of the cell are colored. No, example of a neutral dye is your Jameson stain, which is usually used for staining blood smear. Okay, so let's now proceed now to the different types of staining techniques. Okay, so let's now see the direct staining and indirect staining. So this technique no in staining po usually po employs no the complexity of the procedure done. Now when we say direct staining, this is a process po by which microorganisms are stained with simple dyes such as your methylene blue. On the other hand, when we say indirect staining, now the process po needs more than or when we say more than po these are chemicals that strengthens the binding of your dye or stain on the bacterial cell wall. So your mordant po helps make dye return, no serving as a link or bridge to make the staining recline possible. So it forms a colored lake, no, which in turn no combines with the microbial cell to form a cell mordant dye complex. Your mordant class, please take note, it is the integral part of the staining reaction itself. So without mordant, no staining could possibly occur because your mordant po links the tissue to the receptors no in your tissue to the dye itself. And then without the existence of mordant, you cannot attach permanently the dye to the bacterial tissue. So usually your mordant is applied before the stain or it may be included as part of staining technique or it may be added to the dye solution itself. Now your mordant can some mordants also can act as an accentuator. So which is not an essential to the chemical union of the microbial cells and the dye itself. So usually accentuators po does not participate in the staining reaction, but they merely accelerate or hasten the speed of the staining reaction by increasing the staining power and selectivity of the dye. So you can take it as accentuators are usually po increases no or intensifies the color reaction of your dyes to the bacteria. So proceed now to staining techniques, usually that has basis po when it comes to the procedure itself. So firstly po is your progressive staining. Now when we say progressive staining class, the microbial cells po are stained in a definite sequence. Now in order that a satisfactory differential colorization of the cell may be achieved at the end of the correct time with staining solution. So when we say progressive, so it has a step-by-step definite sequence to achieve differentiation of the cells you want to identify. Now, on the other hand, regressive staining po is done po by overly staining po the whole smear or the whole microbial cell to obliterate the cellular desires. And then the excess stain po are decolorized from unwanted parts. So usually regressive staining po employs excessive or excessive stain and then you decolorize it from the unwanted parts for you to differentiate the cells that you want to identify. So continuing with the regressive staining is the differentiation step. So your regressive staining po employs a differentiation step or decolorization step. So this is selective removal of excess stain from the tissue during regressive staining so that the specific substance po may be stained differentially from the surrounding cell using another type of stain. So usually it is controlled visually by examining it under the microscope and adjusting the type of stain or the contact time of the stain and the procedure itself. Now, these are the uses po of differential staining method. So firstly, it is used to observe the morphology, size, and arrangement of bacteria. It differentiates also one group of bacteria from another group. And then lastly, biological stains are dyes used to stain microorganisms. Now, in terms of number of stains used in the procedure, we can classify your staining method as to simple, no, and then um, differential. So when we say simple staining method class, it only uses one type of dye. So it uses single stain po. And usually po it is usually used po to demonstrate bacterial morphology and arrangement, and it is directed towards coloring the forms and shapes of the cell. Now there exist two kinds of simple stain. This is your positive staining and negative staining method. Now the positive staining method po allows the colorization po of the bacteria, and the other parts po are stained by the dye. And usually the background of the bacteria is colorless. So examples of positive stains used in positive staining are your carbofluoxin stain, methylene blue stain, and your crystal violet stain. Now, on negative staining method po, the dye naman po or the dye po stains the background and the bacteria remains unstained. Now, please take note that the simple staining method, for you to observe a proper um, staining result, there exists po a standard inoculum size. Now, the standard inoculum size in simple staining method is 100,000 colony forming units per ml. So, ibig sabihin na po, you need to have that um, specific type or specific count of bacterial cells for you to observe properly your simple staining method. Now, in this slide shows you the result of a positive staining and negative staining. So please take note, for positive staining, you color the cell, not the background. In contrast naman, negative staining po, you color the background, not the cell. So in this slide shows you the procedure of your simple staining method. So firstly is you make a smear and label it. Then you allow to dry po the smear. Now fix the smear over a flame and then apply a few drops of positive simple stain like your methylene blue, carbofluoxin, and gentian violet for 1 minute. Now after the contact time, you wash off the excess stain with water and then air dry and examine the preparation under oil immersion objective. Now, in contrast with simple stain po, the differential staining method po uses multiple stain. Usually, it uses two stain po, no, to differentiate groups of bacteria or separate groups. So again, it is used to distinguish different cell structures and cell types. And then it is directed towards coloring po the components of the cell. So not the size, no, and morphology of the cell. Okay? So the required inoculum size po for differential staining is the same for your simple stain. You need to have 100,000 colony forming units per ml of inoculum in your preparation. So examples of differential staining methods are the following. These are your gram staining and acid-fast bacilli staining. So I will do no separate discussions for these two important differential staining methods. Now, the basic steps for differential staining would be the following. So, firstly is the application of the primary stain. So, usually primary stain are cationic stains po, used to stain the first structure or layer of your bacteria, which is the cell wall. Now, on the second step is the application of the mordant. Now, the mordant is an important reagent po here because your mordant increases the binding of the primary stain to the cell wall. Now, the third step is the application of the decolorizing agent. So this decolorizing agent will remove the excess um, non-permanently attached dye or stains in the cell wall, and thereby doing the differentiation part. Now, on the last part naman po is the application of the secondary stain or the counter stain. So this will stain naman po other groups of bacteria and then allowing you on the last or on the result to differentiate the two groups or of bacteria uh, based on the differentiation of their color result. Now, to get an overview po of the um, commonly used no, staining methods and other special stains, no, we will start by discussing them one by one. Now, firstly is your gram staining method, which is developed by uh, a scientist, namely Hans Christian Gram in 1884. Now, most bacteria are differentiated by their gram reaction due to differences in their cellular structures. The most commonly used to differentiate the major groups of bacteria, namely your gram-negative and gram-positive bacteria, is by using this type of staining method. Now, the gram-positive bacteria po are bacteria that stains purple with the crystal violet dye or the primary dye after decolorizing with acetone alcohol. Now, gram-negative bacteria, on the other hand, are bacteria that stains pink with the counter stain or the safranin after losing the primary stain when treated with acetone alcohol. Now, the required reagents for gram staining method are the following. Firstly is your primary stain, your crystal violet, your mordant, which is your grams iodine, your decolorizer, which is your acetone alcohol, and lastly is your counter stain or the secondary stain, which is safranin. Now, quality control organisms are used to assess whether your procedure is accurate po um, in releasing a result. Now, the quality or control organisms used for gram staining methods are the following. Now, for the gram-positive bacteria, we use Staphylococcus aureus with an ATCC number 25923 for gram-positive result. For gram-negative bacteria naman po, we use Escherichia coli with an ATCC number 25922 for the gram-negative reaction. Now, the general rule po in gram staining is that all cocci po are gram-positive except for the following. So, your Neisseria, Veillonella, and Branhamella or Moraxella po are the only known significant bacteria po that are cocci but they are gram-negative in nature. Now, all bacilli naman po are gram-negative except for the following. So, these are the gram-positive bacilli, your Actinomyadura, Arcanobacterium, your Bacillus, Clostridium, Corynebacterium, Erysipelotrix, Gordonia, as well as Corthia, Listeria, Mycobacterium, Nocardia, Rhodococcus, Streptomyces, Trophaema whipplei, and Chucamorella are the gram-positive bacilli. And then there are also organisms that cannot be readily stained by gram staining. So these are your chlamydia, your mycoplasma and ureaplasma, and your spirochetes. Now, next important staining procedure is your acid-fast staining. So, unlike gram staining reaction, your acid-fast staining procedure doesn't allow your bacteria to be grouped accordingly as gram-positive and gram-negative. So, your acid-fast staining procedure group your bacteria into acid-fast bacilli and non-acid-fast bacilli. Now, this technique po is applied to bacteria with high lipid content in their cell wall, specifically the mycolic acid po. And then it utilizes po three reagents, no? Your primary stain, your your decolorizer, and your secondary stain. Its model is not a can either be a chemical or a physical method. So usually it utilizes carbulfuchsin as the primary stain and methylene blue or malachite green po as the secondary stain. Now the cell wall of acid-fast bacteria resists the acid alcohol. So we use acid alcohol for decolorization po na step. So pag sinabing resist po, it doesn't allow po the passage po of the acid alcohol in the cell wall of your acid fast bacteria because of the high lipid content. Now, there exist two common methods under acid fast staining. This is your Ziehl-Neelsen method and your Kenyon method. Now, for the Ziehl-Neelsen method and Kenyon method, the only difference of these two is the more than po. So, what type of more than it uses? For Ziehl-Neelsen method, it uses heat, no, as more than, while your Kenyon method naman uses the chemical tergitol as its more than. Now, to summarize again, your acid-fast staining methods employs four steps po, just like your differential staining method na gram stain. So, it employs a primary stain, which is your carbonfuchsin, a more than, it's either heat or tergitol, a decolorizer, which is acid alcohol in this method, and the fourth one would be the counter stain or the secondary stain, which is your methylene blue or malachite green. Now, the quality control organisms for acid-fast staining are the following. For positive AFP, this is your Mycobacterium tuberculosis, ATCC 25177, H37RA. For positive partially AFP, this is your Nocardia asteroides, ATCC 3308. Now, lastly, for negative non-AFP, this is your Escherichia coli, ATCC 25922. Now, as you can see here, I put a color na already on the results po. So as for you to remember no easily the color reactions when it comes to the result. Now, for the acid-fast bacilli, so when we say positive acid-fast bacilli, no, the reaction is usually red or red. Okay, so pag positive ang AFB, it is usually color red in reaction under acid-fast staining. For the partially acid-fast bacilli naman po, when we say partially, it doesn't completely take up the primary stain, no? So usually the result of their color is usually pink, no, or pale red. Now for the non AFB that doesn't take up the primary stain and they usually take up the secondary stain, which is color blue or green. Usually po, they are color blue or green in the result. Now, this table po shows you the summary of the different staining methods under AFB. So, besides the commonly used staining methods, there's Ziehl-Neelsen method and Kinyoon, which is alternatively called as hot staining method and cold staining method. So, there exists also the Pappenheim, no, the Baumgarten, and the Oramin-Rodamin method. So, when we say Pappenheim method, this is usually used to differentiate the other family of Mycobacterium tuberculosis, which is known as your Mycobacterium smegmatis. Now, so your Pappenheim method again differentiates Mycobacterium smegmatis from Mycobacterium tuberculosis. In terms of color class, your Mycobacterium tuberculosis in Pappenheim is color red, while your smegmatis po is color blue. So for your Baumgarten method, it differentiates naman po M. leprae or Mycobacterium leprae from Mycobacterium tuberculosis with the same color reaction as with your Pappenheim method. Now, in your Oramin-Rodamin method naman po, it employs a fluorescent dye to selectively color the AFB cell wall. Now, usually, your acid-fast bacilli po in your Oramin-Rodamin appears as reddish-yellow fluorescence under a dark background. So, it fluoresces as a glowing reddish-yellow bacilli. Now, on the Ziehl-Neelsen method, again, your bacilli is color red against blue background, while in your Kenyon method, your bacilli is red, while your background can be blue or green. Now, besides to the two most commonly used staining method, your gram staining and your acid-fast bacilli staining, so there are also other special stains used to color specific parts of the bacteria. So firstly is your endospore staining method. So it stains specifically the endospores of the certain bacteria that can produce spore po. Okay? So the different methods to use are the following: your acetic acid method, which contains carbonfuchsin, acetic acid, and your Loeffler's alkaline methylene blue for the secondary stain. Now, the most commonly known naman po na endospore staining method is your Ritz-Conclean or your Schaefer-Fulton method, which employs malachite green as the primary stain, and then its secondary stain naman po would be saffronin red. Now, its more than po is heat, just like your AFB staining method. Now, lastly is your Dorner method, which employs the carbulfoxin as its primary stain, and secondary stain would be your nicrosin dye. Now, in some cases po, we can also stain the capsule, no, of certain bacteria that produces capsule, no. Now, these are the examples of capsule staining method. So the most commonly known is your His method or Anthony's method. And then for Novelli's method naman, it uses a specific type of dye which is known as your alkyan blue, no. And for your Lamb method, it uses one type of dye, which is your Loeffler's alkaline metal blue. And lastly naman po would be your Nigrosin method, which uses one type of negative stain, which is called Nigrosin dye. Now, in some cases naman po, we need to demonstrate the presence of a type of appendage in your bacteria called flagella. So flagella is used by certain bacteria um, to have a certain type of motility. Okay, and in some cases we need to demonstrate what type of flagella are they have. Okay, and then we usually observe it using staining methods. And the staining methods for flagellar staining are the following: your gray method and Leifson method. For gray method, it uses carbulfoxin and tannic acid for mordant, and for your Leifson method, it uses carbulfoxin, tannic acid, and additionally it uses methylene blue as its secondary stain. Now, in this table po shows you the commonly used staining techniques, no, and its employed stain and the cellular structure or bacteria it stains. And I also included here are other stains for other cellular structures such as your metachromatic granules, the cell wall, no, the DNA, and also to certain bacteria like your spirochetes. So please take note now. Classic examples po of staining techniques that colors the metachromatic granules in your bacteria is your Albert and Neisser. Na examples naman po of stain used for spirochetes, a special type of spiral bacteria, is your Fontana tribundu, your Levaditi po, and your um, that too. Okay, your Fontana tribundu and your Levaditi. So example naman po of a DNA stain is your Felguen or Fulgen stain, which is it contains lang po carbonfoxine. Okay? So please take note for your spirochete stain, your Fontana Taibundu po and Levaditi. It uses silver compounds or metal-based stain like your ammoniacal silver nitrate and your tannic acid as its mordant. Okay? So another type of um, stain is your Dyer stain. Your Dyer stain naman po specifically stain the cell wall of any type of bacteria which contains po the Congo red. Now, lastly, under special stain is using naman po a very specific method using a diagnostic antibody or a DNA probe mediated staining. So when we say antibody na po, we usually attach the stain to an antibody to specifically color a part or type of organism. So it is used for the specific identification of selected pathogens such as your Chlamydia trachomatis, your Bordetella pertussis, and Legionella pneumophilia. It can also be used by demonstrating certain types of viruses, like for example, your human simplex virus, your varicella zoster, your cytomegalovirus, adenoviruses, and respiratory viruses. Now, to summarize with all of the discussed stains, no, I have included you a simple table, no, that summarizes po the different stain type, the specific dyes used, no, the purpose and the outcome, and the sample image po for each type of staining procedure. For basic stains po, usually we employ lang po specific dyes or basic dyes like your methylene blue, crystal violet, and others. And then the very purpose of basic stains are to stain, no? And demonstrate po the simple structure and morphology and arrangement of your bacteria, but do not differentiate groups. Acidic stains naman po, examples of these are your eosine, acid fuchsine, rose bengal, and Congo red, and they stain positively charged molecules. Okay? Negative stains naman po, no? Stain po the background, not the object or the bacteria po. And examples of negative stains are your India ink and Negrosin. Now, for gram stain po, this is a type of differential stain that differentiates the two major group of bacteria, namely your gram-positive and gram-negative bacteria. Now, the outcome would be, now if the bacteria stain purple, it is considered as gram-positive. Then, if the bacteria stains pink, it is called gram-negative. Now, in the acid-fast stain, so usually po it is a type of special differential staining method which stains po the cell wall of the bacteria with high fat content, okay, or with high mycolic acid content, such as your mycobacteria family, okay. So the outcome of the acid fasting would be if the bacteria is in red in the end of the reaction or the procedure, you can identify it as an acid fast bacteria. On the other hand, if the bacteria is color blue, no, you will report it naman as non-acid fast cell. Now, for your endospore po, specifically endospore stains, no, it stains naman po the spore content or the endospore content of certain bacteria. Okay? And then the widely known method would be your Schaefer-Fulton procedure. And the outcome usually po is that the endospores appear as bluish green and the other structure of the bacteria appears pink to red, as you can see here in the picture. So if the bacteria has green in the inside, that means po it has endospore. While if it doesn't have the green staining result at the end, the bacteria po doesn't have endospore, do not produce endospore. Now, lastly is your flagella and capsule stain. Now, for your flagella, again, no, usually the flagella po, that's why other methods po on the flagellar stain uses tannic acid or potassium alum because this serves as the mordant, no, it coats the flagella, no, no, thereby allowing po the attachment mo of the dye, no, that you will employ to the flagella itself. Now, the flagella usually are visible if present. So, as you can see here in this picture. So those string-like appendages in your bacteria are the flagella. Now, lastly, in the capsule stain, it stains naman po the capsule produced by bacteria. Okay? And usually, we employ negative staining techniques to demonstrate capsule formation. Okay? So it distinguishes cells with capsules from those without, and the result would be your capsules appears clear or halo, no, if present. So that would be the end of our discussion. Thank you everyone for listening. Have a good day.